Well, now we are starting Mendelian genetics, and this is the chapter outline. So, we are going to see history, the choice of experimental material, method, then the principles established, illustration, that is example, and where it is applied, and what are the exceptions. Here we start with the history part, right? So when we come to the history part, and here you don't have to really write down, okay? You don't have to copy it with me. Try to understand if there is anything to be written down. I will specifically tell you that you please write down, and I'm anyway going to upload this presentation also. And the presentation or the PDF, neither of them have to be verbatim copied at all. That will be a sheer and very bad wastage of time. At the end of it, I will be giving you a question, and what you are supposed to do is to combine my lecture notes or lecture presentation, this one, and my PDF to answer those questions in an outline form, not the full and formal kind of answer. So oftentimes we tend to think that the full name was Gregor Johann Mendel. Mendel was born, raised, and he resided in that part of Europe where this spelling does not get pronounced as Johann, it is pronounced as Johann. So his real name was Johann Mendel, and Gregor was the title, Gregor was the position that he held in the St. Thomas's Abbey. So that is where he was there. So that was a monastery, and in the garden of the monastery, Mendel, just out of his love and curiosity for the natural world, he decided to follow the pattern of inheritance. Interestingly, Mendel did not work with just garden peas. Mendel also worked with a number of other plants. But for variety of features, garden peas proved to be a little superior choice of material. We are going to see that. So it was easy to work with garden peas. It was easy to analyze the results of inheritance pattern of different characters, and that is why Mendel chose to stick with garden peas. So if you ask me, did Mendel work only with garden peas? Answer is no. Mendel worked with Vicia faba. Mendel tried to work with Lathyrus odoratus, and of course Pisum sativum. He stuck with Pisum sativum, garden peas, because there were a lot of advantages, especially with respect to the structure of the flower, which allowed Mendel to easily carry out crosses. I'll show you how. And that is why Mendel stuck with that. His experiments were conducted over a period of almost 7 years. Between 1856 and 1863, during this period, Mendel studied the inheritance pattern of certain traits in garden peas, and obviously in these many years he would have worked with a very large number of plants and across several generations. And then in 1865, 2 years after he stopped conducting his field work, 2 years down the line, in the local Natural History Society, Mendel for the first time presented his findings. Scientifically, we call it reading the paper. And a year later, Mendel published his paper, the paper that he had presented in the local chapter of the Natural History Society. So a year later, the same paper with certain more polishing and all got published. So the year of Mendel's works publication is 1866. Of course, the title of the paper was Experiments on Plant Hybridization. You are not supposed to remember the German name that was there, of course. And paper was presented at the Natural History Society in Brno, and that is where Mendel was a monk in the monastery in 1865, and the very same findings were published a year later. You don't need to really remember the name of the publication, it will be very hard for us to remember. So don't even burden yourself. So that is the history part. And like I said, that almost for 35 years minimum, the work of Mendel was not noticed by the scientific community. And then three different scientists, Schermark, Hugo de Vries, and Karl Correns, independently in the year 1900, they rediscovered the paper that Mendel had published. Not only did they rediscover, but they repeated the experiments. First, they repeated the experiment on garden peas so that they can validate by themselves if the findings of Mendel were really what he reported. Second, based on these findings, they tested the principles of Mendel, that is segregation, dominance, and independent assortment, and they themselves tried to see if the findings, if the numbers are actually being explained by the three principles given by Mendel or not. So they did that as well. And then they repeated the same set of experiments on different plants, different animals. And this is where what happened was that they did not find the same pattern in certain species. And that led to discovery of linkage, that led to the discovery of cytoplasmic inheritance and sex-linked inheritance. So wherever the laws were found applicable, so they validated the Mendel's laws, and wherever it was not found to be applicable, there were such cases where it was not found to be applicable, but the good thing was that it led to discovery of other phenomena. So that ways, in the field of genetics, these were very exciting years. The first 50 years of the 20th century have actually been very, very exciting years for the field of genetics. So many developments took place. For example, rediscovery of Mendel's work, chromosome theory of inheritance by Sutton and Boveri, proper discovery of linkage, and then its proper explanation by two different scientists. Linkage was discovered by I'm sorry, I'm forgetting the name. So linkage was discovered, and a little later, linkage was properly explained by Thomas Hunt Morgan. Around this time, a variety of sex determination mechanism, they were also discovered. Well, linkage was discovered by Punnett. Punnett was, R.C. Punnett was an important scientist of that time. So Williamson and Punnett, they discovered the phenomenon of linkage, and its explanation came from the work of T.H. Morgan. Then sex determination mechanism, Frederick Griffith's experiment, then one gene, one enzyme theory. Then DNA was discovered to be the genetic material. Then the double-stranded DNA structure was elucidated by Watson and Crick. All of this happening in approximately first 50 years of 20th century. So well, right now coming back to Mendel. So Mendel's work got not only rediscovered, but revalidated, and we understood what was the contribution of Mendel. And that is why Mendel is correctly called the father of modern genetics. When we say modern genetics, we basically mean genetics that is rooted in science, not only in hypothesis and imaginary explanations. Now we are coming to the experimental methods of Mendel. So when you come to experimental methods, here we are not going to discuss what caused the success of Mendel. Here we are going to discuss why did Mendel choose garden peas as his experimental material, experimental plant. And second, we are going to see what was the design of Mendel's experiment. So first and foremost, let's see why did he select garden peas. 6 very clear advantages. First and foremost, garden peas had many variants and contrasting character. Like if you take the height of the stem, so garden peas has got two contrasting types, tall stem, dwarf stem. If you take the color of the seed, two contrasting varieties, yellow colored seed and green colored seeds. If you take the pod of the seed, again color could be yellow or green. Similarly, if you see the shape of the seed, it could be full or it could be wrinkled, round or wrinkled. So similarly, the color of the flower, it could be purple or it could be white. So there were so many variants, and these variants which Mendel had chosen, he took 7 different characters, and for each character, Mendel was able to find 2 clear-cut contrasting types. So that was one thing. And then Mendel could also establish that these variants were genetically pure. What do we mean by genetic purity? We mean that if the tall plants are continuously mated with other tall plants, then for any number of generations, they will give only the tall plant. That will mean that they were pure tall plant. Similarly, if the dwarf plants are mated with dwarf plants, for any number of generations, they will keep producing dwarf plant. As a matter of fact, dwarf is recessive, and that will anyway be true breeding, but tall may or may not be true breeding. Okay. And nevertheless, Mendel spent almost 1 year's time out of his 6 years in establishing the pure line for tall, pure line for dwarf. Mendel at that point of time was not aware which is dominant, which is recessive. These were the terms which he coined even later, not at that time. Similarly, he created a pure line of purple flowers, pure line of white flowers, pure line of green seeds, pure line of yellow seeds, pure line of green pod, pure line of yellow pod. So that he did. Next, garden peas are very, very easy to maintain. You sow the seeds, you just take care of the plant, basically little bit of watering and all of that, and the plants will be growing very well. So they are easy to grow, easy to maintain. Vivek is asking, true breeding means what? So true breeding, like I said, Vivek, that if it is a tall, wait a moment, if it is tall, so if you cross it with tall, next generation is also going to be tall, next generation is also going to be tall. So that will be true breeding. And same you do with dwarf, that if you cross dwarf with dwarf and generation after generation it is giving you dwarf, that will be true breeding. And you will later realize in this discussion only that sometimes it may happen that it is a tall plant, you mate it with a tall plant, but still it can produce some tall and some dwarf plants. So such a tall plant will not be called true breeding. But if a tall, by getting crossed with a tall, always produces tall, is true breeding tall. So he established the true breeding line, Vivek. And then, like garden peas were easy to grow, easy to maintain, choti life cycle thi. So within a short span of 5 years of serious experimentation, 1 year to he has spent in establishing the true breeding line. बिल्कुल अब्राहम लिंकन की तरह. अब्राहम लिंकन ने कहा था कि if I have got 6 hours to chop a tree, I will spend 1 hour planning how to chop the tree. So Mendel भी उसी मोड में थे कि if I have got 6 years to carry out my experiment, 1 year I will spend in preparing for that experiment. So after that, when he started to grow, so not only garden peas were easy to grow, but they had short life cycle. So short life cycle का फायदा क्या है ना कि you can get many generations. And when you get many generations, so it is very, very obvious that you will have a lot of data to collect and analyze. So short life cycle, quickly you will see first generation, then second generation, third generation, and you can keep validating your data. Then Garden peas, मटर ही तो होता है ना. So garden peas have large number of offsprings. Just think, within a single pod, right? Within a single pod, how many seeds do you normally find? Depends, but anywhere between 6 to 8 seeds you are going to find. The question is how many such pods you will be finding per plant. And now you can guess what will be the number of offspring. The greater is the number of offsprings, the more accurate your data analysis will be. The lesser is the smaller is the sample size, the more error prone your data will be. Let me give you this example. The example could be like in this class, apart from me, there are right now 43 students. Let me ask, what is your optional subject? You will think, why am I even asking this question, right? So answer is obvious. Zoology की class है, तभी तो class में बैठे हुए हैं, okay? But for the sake of asking this, let me ask this, and all of you will be answering zoology. And then, what if I get this conclusion that everyone preparing for civil service is preparing it with zoology? So will it be the right conclusion? Answer is no. Where did I get wrong? I got wrong because in the beginning I didn't know who's having what optional. So the only thing that went wrong was my sample size was too small. If I speak with, rather than 43 students, I speak with 43,000 students, for that matter even 430 students from different locations, and I will have very different answer. But if I ask this audience only, you know what will be the outcome, right? Now, Nitish is saying that this will also produce tall. It will produce tall, but not generation after generation. You carry out selfing of this tall, and in the very next generation, you will see dwarfs also arising, and that will mean that it is not pure breeding, Nitish. I hope you understood my point. And sixth point is flower structure is easy to manipulate. Basically easy to manipulate क्या होता है कि जब भी आप किसी दो प्लांट को क्रॉस करते हो, हाइब्रिडाइज करते हो, सो आप इस वेराइटी की पॉलन उस वेराइटी पे you will land. Basically, in hybridization, let's say that you are hybridizing a tall plant with a dwarf plant. So you will take pollen from tall plant and you will put on the stigma of dwarf plant. And then you will see what the next generation is going to be like. So whenever you do this, it is important that the dwarf plant's stigma does not get pollinated by the pollen grain of its own flower, because garden peas have got complete flower. Complete flower means it will have a stamen and it will have carpel also. So while you are getting the pollen grains from the tall one, what if pollens from here come and cause pollination? So then dwarf get pollinated by its own pollen grain, and then it will be producing dwarf plant. So the flower structure should be such that you should be able to cut out the stamen, and you should be having the liberty to introduce the pollen grain from other variety. And somehow garden peas had the flower structure which was really very easy to manipulate. Even in the young stage, जब pollen बना भी नहीं था, Mendel was able to manually, with help of a scissors, cut away the stamens, and that way cell pollination was prevented, right? So these were the 6 scientific reasons why Mendel persisted with garden peas. Like I said, he started with other plants also, but he persisted with this one. His design of experiment was remarkable, especially for the time when the experiments were happening, 1856 to 1863. And then Mendel decided to focus on a limited number of characters at a time. So at a time, Mendel focused on one character, that is monohybrid cross, 2 characters, dihybrid cross, and Mendel also carried out limited number of experiments with 3 characters at a time, but Mendel did not go beyond that. And Mendel, in his first paper, did not even mention the results of trihybrid cross. Mendel stopped at dihybrid cross, and with help of that, he tried to explain the theory of the principle of independent assortment. So when he focused on a limited number of traits, it became very easy to analyze the data. The next one, Vasvi Chaturvedi is saying, please tell once again what is a complete flower. A complete flower, Vasvi, is a flower that has got a stamen and carpel together within the same flower. So that kind of flower will be called complete flower. So garden peas have got complete flower, but thankfully the structure of the flower is such that you can remove the stamen. I hope it is clear, Vasvi. Then Mendel was able to identify the contrasting varieties for those character. Character is height of the stem, and then the contrasting varieties will be tall dwarf. The character can be color of flower, and the varieties can be purple and white, so on. And then he went on to establish the purity of varieties. I have already spoken about the true breeding thing. And this is something which none of his predecessors had attempted to do. And then Mendel went ahead with hybridizing the contrasting varieties. Well, that will mean tall with dwarf, green with yellow, so on, round with wrinkled, and all. And then Mendel analyzed the progeny, counted the numbers, and kept a proper statistical record so that he can later analyze. And after analysis, point number 6, Mendel formed hypothesis that, okay, if these are my findings, what explains these findings? And then he did not stop there. If this was his hypothesis, he tested the hypothesis. Means he carried out more experiments. Let's say that on the round versus wrinkle shaped seed of the shape of the seed, he carried out experiment. And he came up with a conclusion of dominance. Then he tried to repeat it on tall and dwarf, green and yellow seed, and so on, so that he's testing the hypothesis. And once his hypotheses were validated, he derived a principle, that this is a principle. Such was the meticulous approach Mendel followed. So was Mendel lucky? A little lucky, yes, because the crosses that he performed, out of 7, 6 of them were unlinked. The 7th one was linked with the 5th, but somehow Mendel did not report that. So a lot of people say that perhaps Mendel did not publish that data because it did not fit in the framework. Whatever the truth is, we can only guess. Whether Mendel conducted that experiment and then did not report the finding because Mendel was unable to explain, or Mendel did not conduct the experiment, whatever the case was, but there was a tiny amount of luck. But it was only tiny amount of luck. No amount of luck will be substituting the 8 things that we have just now mentioned. So that is how Mendel conducted his experiment. Now we are moving to the structure and manipulation of Pisum flower. So Mendel removed the stamens of one type of flower, for example here the purple flower, and then he transferred the pollens from stamens of white flower. So first and foremost, with the help of scissors, you cut the stamens. Let the carpel be there, let the female part be there, and now female part has no chance of getting pollinated by its own, the same flower's pollen. And then he brought pollen from the stamens of white flower to the carpel of purple flower. And here you go, you have introduced the reproductive propagule from the other variety. Now it will be interesting to see what happens to the next one. So then he pollinated, the pollinated carpel matured into a pod. Then Mendel planted the seeds from the pod, and then he observed the traits of the offspring, that now if the offsprings are having purple flower or white flower. And then Mendel repeated it by causing these offsprings mate among each other. That is called selfing. This was cross. This was crossing because pollen is moving from one type to another type. And this is called selfing because pollens are moving among the same type of plants. So first parents were crossed, and then the first generation F1 were self to generate F2, and we are going to see what happens then. Right? So this is how Mendel manipulated the flower. Now, these were the 7 characters Mendel studied. It is there in your PDF as well. The flower color, at that point of time, Mendel was not aware what was dominant and what was recessive, but of course, when he conducted the experiment, it became clear what was recessive and what was dominant. So, purple flowers are dominant, recessive is white. The position of flower, if it takes a place laterally on the stem or the branch, that is called axial, that is dominant, and terminal, that is at the tip of the stem or the branch, that is recessive. Yellow seed color is dominant and green seed color is recessive. Round seed color shape is dominant, wrinkled is recessive. If the pod is inflated, it is dominant. If the pod is constricted, it is recessive. If the pod is green, it is dominant. If the pod is yellow, it is recessive. And if the stem length is tall, it is dominant. And if the stem length is dwarf, it is recessive. So that's how Mendel carried out these 7 characters analysis. And mind it, each character has 2 types, 2 opposing phenotypes, 2 opposing traits, that is contrasting features with respect to the same character. So character is this, these are traits. Okay. And now couple of terms before we move further. Okay. So gene is that genetic factor, that genetic information that helps to determine a heritable characteristic. Allele is an alternate form of a gene that is placed on the same locus. And what's locus? Locus is the location of the gene on a given chromosome. Genotype is what is your allelic combination. Because remember, any individual who is diploid will have 2 sets of chromosomes that will mean 2 copies of every gene. Now, the 2 copies of the gene can be identical. In that case, the individual will be homozygote. And if 2 different alleles are present for the same gene, then the individual will be heterozygote. Phenotype is what kind of manifestation of a character we are seeing. That is phenotype. For example, for height, which is a character, what is the phenotype? So it is tall or dwarf. For flower color, that is a character. What is the phenotype? It could be purple or white. So that ways. Okay. And then here, any diploid individual, including we the human beings, will possess 2 sets of chromosomes. That is why we human beings have got 46 chromosomes, 23 coming from mother, 23 coming from father, right? And father will give us 1 set of gene copies and mother will give us 1 set of gene copies. Therefore, every single gene that we have got has got 2 copies, right? Minus the genes located on X and Y chromosome. So in mammalian females, the genes which are located on X chromosome will be present in 2 copies, but in mammalian males, the genes present on X will not be present on Y. So, well, that is a difference. But for all other chromosomes, that is 22 chromosome, it will be correct to say 22 pairs of chromosome. And all the genes which are present on these 22 chromosome, there will be 2 set of 22 chromosome. Therefore, there are 2 copies of each of the gene that is present. So approximately 24,000 genes are there in the human beings. So we have got 2 copies. But those 2 copies need not be identical allele. If there is an identical allele present in the individual, both the copies are the same, that will be homozygous for that particular gene. And if the two alleles are different, then the person will be heterozygote again for that particular gene, right? So here we are. This is the gene locus, and here the person has got one dominant allele and one recessive allele. So this plant is basically heterozygote. Any question until now? Until now it is pretty much NCERT level stuff, but still if you have any question, please raise your hand and I will take your question. Okay. And now we are moving to the easier one of Mendelian cross, that is the monohybrid cross. So in monohybrid cross, what Mendel did was to cross two varieties, two contrasting varieties of a single character. So that is what Mendel performed. And Mendel performed this analysis for two subsequent generations. So first beginning will be made with parents, and the first generation of offspring will be called the F1 filial 1. And then the second one will be called F2. So two parents will be crossed. They will be contrasting for the same character. So tall dwarf that way. And then whatever is F1, that F1 will be that all the F1 individuals will be selfed, means they will be self-pollinated. And then we are going to see the result of F2. How does F2 show up? And mostly we have seen that in F2 the contrasting characters resurface. In F1 only one of the trait will be shown. Let's say that when you cross tall plant with dwarf, so in F1 all are going to be tall, but when you carry out selfing, so you are going to see tall in proportion of 3 and dwarf in proportion of 1. There is a question, let me see that question. Okay, so in F2 you will see again contrasting traits coming up, but in F1 one of the contrasting trait will show, and that will be basically the dominant trait. So let me see what is the question and who it has come from. Kajal Kumar Yadav, sir, you told something about mammalian male and female. I just got confused. Okay, so I was basically saying that mammalian males have got 22 autosomes in 2 copies. Okay, plus mammalian males have got X and mammalian males have got Y. Whereas mammalian females have got 22 copies of autosomes, 2 sets, plus they have got 2 copies of X chromosome. So here you can say that in mammalian females, they have got exactly 23 chromosomes in 2 sets, because even X is present in 2 copies. But in mammalian males, you have got 22 chromosomes in 2 sets, plus X and plus Y, because the genes which are present on X are not present on Y, and vice versa. So that's what I said, Kajal. Okay. So I hope my speed is okay. It is not too fast or too slow. There can always be opinion, but I hope you are comfortable with the speed. Okay, thank you. Chalo. So Mendel first carried out the monohybrid cross, and you know what is a monohybrid cross. So monohybrid cross is essentially one character, 2 contrasting traits, and in generation 1, F1, you will find one of the traits. That trait will be dominant. Here we go. So first the experiment here, and then the explanation here. And when in the examination you are writing, you don't really have to draw these purple colored flowers and white colored flowers. You don't have to do that. You can, if you are talking about phenotype, you can simply talk purple, white, that way. And if you are talking in terms of genotype, you can write like this. But let's see this one. So here. So Mendel got pure purple flowers, which will only get purple if you are continuing to mate them with purple, and pure white flowers if you continue to mate them with white. Mendel crossed them. And Mendel was so wise that he performed reciprocal crosses. What do we mean by reciprocal crosses? Let's say that if in this cross, purple flower is the donor of pollen and white flower is the recipient of pollen, then Mendel set up another experiment where purple flower was recipient of the pollen and white was the donor of the pollen. Can you imagine that in 1856 Mendel performed such meticulous experiments? And then Mendel saw that in both the cases, whether purple is the male partner or purple is female partner in other set of experiment, in both the cases, the F1 generation, the hybrids, were having all purple flowers. This kind of experimental setup is known as what? Reciprocal crosses. And Mendel was careful enough to carry out reciprocal crosses. But well, when these flowers were crossed with white, in the F1, all the plants had purple flowers, right? And then fertilization among the F1 plants. So this is called selfing. So now the purple flowers which have been generated in this way, so they will be selfed, means among them there will be self-pollination. And then what you see? You see that 3/4 of the plants display purple flower and 1/4 of the plants display white flowers. And then Mendel went ahead to explain. What Mendel said, that for any factor there are different alleles. Alleles are alternating form of factors. Mendel used the word factors for the genes. The term gene got coined in 1902 by Wilhelm Johansen. Before that, the word was factor. So Mendel said that for any factor, there are two different types of allelomorphs. And in the parents, there are two copies of the factor. The pure parents will be homozygous, so it has got one type of allelomorph and it has got other type of allelomorph but same type because they are pure. Mendel also said that at the time of gamete production, the two copies of the factor will separate into one copy. So gamete will carry a single copy of the factor, whereas the adult individual will be carrying two copies of the factor. So separation of the two copies of the factor, whether they are homozygous or heterozygous, so separation of the two copies of the factors so that in the gamete a single copy of the factor is present is known as segregation. So when for gamete production, when for gamete production you have got separation of the factors, this is known as segregation. What would have happened had this been heterozygote? Then one set of gamete would have had this allele, other set of gamete would have had this kind of allele. This phenomenon is called segregation. Remember, at the time of Mendel's working, nobody knew about meiosis, during which the homologous chromosomes are separated. But Mendel was able to hypothesize, right? Mendel was able to hypothesize about the factors being separated. So Vasvi, I was saying that let's say the parent has got, your question is, I'm reading out the question first. Sir, please explain one factor for one gamete again. So Vasvi, see, this is the parent. Parent has got two copies of the factor. But when this parent produces the gamete, so gamete will have only one copy of the factor And why the gamete will have only one copy of the factor? Because when the gamete fuses with other gamete, then in the zygote, again, two copies of the factor will be restored. If you don't do that, then with every generation, if you don't do that, then with every generation, the number of gene copies will keep increasing. Let's say that there is no segregation, then in the gamete, it will be like this, and in the gamete, it will be like this. Then what will happen in the next generation? Many more sets of genes, and that will be totally unmaintainable. So I hope you have understood that at the time of gamete production, the factor copies separate from each other. This is known as segregation. And Mendel could hypothesize even before the scientific world could ever come to know about something known as meiosis. Tab bhi Mendel could say all of these things. Now, this gamete unites with this gamete, correct? Both these gametes have united, and then the next generation is heterozygote. So it has got one of this allele, one of this allele. And they all had purple. And that gave Mendel an idea of dominance. That when a given factor has got two different alleles, in the heterozygotic condition, one of these two alleles will control the phenotype. For example, in this case, this one has controlled the phenotype because this one was making the flower purple, and because one copy of this is still there, therefore all the flowers are purple. So Mendel said that among the two allelomorphs, one of them will be dominant because in a heterozygotic individual, this allelomorph will be controlling the phenotype. So from monohybrid cross, Mendel got the concept of segregation, that is separation of the two copies of the gene, and he called it factor. And second, dominance, when he saw heterozygote showing one parental phenotype rather than both the parental phenotype. Okay. Shraddha is asking, why are we calling two copies of the factor, as factor stands for gene? So shouldn't we use allele? No, Shraddha. We should still use the word factor. I'm very careful about my words, so I don't say them without a particular meaning. So when two copies of this particular gene is there, so we will call it two copies of the gene only. Okay. And now these are two different alleles. So allele is what? Different variant of the same gene. So even in this individual, even in this plant, there are two copies of the gene. Even in this plant. But these two copies are different from each other. Right? And in this case, there are two copies of the gene, but both the copies are identical. So this is homozygous and this is heterozygous. So every individual does carry two copies of the gene. If you are diploid, you will carry two copies of the gene. I hope you got it, Sudha. And I have also defined what is allelomorph. So we are going to use two copies of the gene only, okay? And if these two copies are different, then they are allelic to each other. If they are identical, they are just identical. And then, let's see. So in this case, all of them have got heterozygotic condition, dominant and recessive. Now we can call dominant and recessive because it is now established. So half of the gametes will have the dominant allele, half of the gamete will have recessive allele. Now see. So from one parent, dominant allele, recessive allele came. From other parent, dominant allele and recessive allele came. So just unite the gene copies. So here, two copies of dominant, purple. One copy of dominant, still purple. Remember, it is dominant. And then here, one copy of dominant, again purple. And here, both the copies of recessive. Therefore, it is white. So what is the ratio? 3 out of 4 is purple, 1 out of 4 is what? White. So this is the outcome, and one monohybrid cross proper analysis resulted into what? Discovery of two things: segregation and dominance. Another question has come. Let me answer this question. Sir, the conclusion for the dominance inferred from crosses only from breeding till F1 generation. So, no, in F1 generation, you are a little confused. But when you carry out selfing, then you suddenly see that the white flower character had not gone away anywhere. If you only see purple flower, so right now you won't be able to conclude what has happened to white. Is the purple one masking the effect of white, or has the white allele just perished away, has just vanished away? You don't know. But when you carry out selfing and then you see white reappearing, then you come to the conclusion white was always there, but it was dominated by the purple one. Therefore, you need to go until F2, else you will not be able to call anything dominant. I hope, Vivek, that answer is clear. So now we are here that in F2 of monohybrid crosses, this is the phenotypic ratio 3 versus 1, and this is the genotypic ratio 1 to 1. Two are heterozygote, then one is homozygote dominant and one is homozygote recessive. So genotypic ratio is 1 to 1, which is given here, 1 to 1, and this is 3 versus 1 is the phenotypic ratio. Now that we have understood this, so we are now ready to take our journey to the dihybrid crosses. So here I have just defined segregation. So segregation is separation of alleles at the time of gamete formation, and it is followed by random fusion of the gametes to produce the zygote. And dominance is out of the two alleles, a particular allele governs the phenotype of the heterozygote, and that allele will be called the dominant allele. And now we turn our attention to the dihybrid cross. With dihybrid cross, we mean we are now focusing on two characters and their contrasting varieties. Let's say the two characters are height of the stem and shape of the seed. Okay, so here the contrasting traits will be round and wrinkled, and here it is tall and dwarf. And now that we are familiar with what is dominant, what is recessive, so we can very well say that tall is dominant, dwarf is recessive, round is dominant, wrinkled is recessive. So what Mendel did, Mendel picked up tall plants with round seeds. So that was one, means combination of both the dominant, and Mendel crossed it with dwarf plants with wrinkled seeds. And then Mendel saw that in F1 all of them were tall and round. All of them were top and round. And then Mendel went for selfing. And selfing led to F2, which has famous phenotypic ratio of 9:3:3:1. So 9 is where both the dominant trait will be seen. That will mean 9 out of 16 plants will be still tall and will still show round seed. 1 will be recessive, means 1 out of 16 plants will be dwarf and they will produce wrinkled seed. But these were unique ones. So these 3 out of 16, 16 is what? 9 plus 3, 12, plus 3, 15, plus 1 is 16. So this one is recombinant because here the plant is tall but seed is wrinkled. And this is also recombinant because plant is dwarf but seed is round. So that Mendel got. And this ratio could be possible only when the allele for height is segregating totally independently from alleles for the shape of the seed. And that gave Mendel an idea into independent assortment. There is a question I will have to take. Is it that only monohybrid cross leads to law of segregation? No. Like Malvika, let me put it a little differently. Monohybrid cross is enough to establish that segregation happens. Segregation you can prove also using the dihybrid cross, but you really do not have to go to that level. Monohybrid crosses will be enough to prove that the segregation, that is separation of the gene pair, is happening. The gene pair can be same allele or different allele. Is it clear, Malvika? Is it clear? और if there is any question, okay, ठीक है, clear है तो बहुत अच्छा है. अब देखो. So here, round and yellow seeds. So pure breeding Mendel took. Pure breeding means homozygous for round, homozygous for yellow. And wrinkled and green seed, homozygous recessive and homozygous recessive. Now the gametes were produced. So one copy of the shape gene, that is round, one copy of the color gene, that is yellow, and then this one will be giving rise to one recessive copy of R and one recessive copy of Y. Fertilization will happen and F1 is going to be what? All the F1 is gonna be the heterozygote, that is one copy of round dominant, one copy of recessive, one copy of yellow dominant, and one recessive. So they all are going to display round and yellow because why? You have got one dominant copy, so dominant trait will be visible to you. I hope this much you understand. Now they are producing the gametes. Okay. Now look at the gametes they have produced. So in this gamete, dominant R and dominant Y are coming together. That is pretty much like the parent dominant R and dominant Y. In this one, recessive R is coming with recessive Y, pretty much like the other parent. Perfect. But was there any parent who produced this kind, where with dominant R, recessive Y came? No, there was none. So how could this happen? This could happen only when segregation of R has nothing to do with segregation of Y. This is called independent assortment. And without independent assortment, you are never going to get 9:3:3:1 ratio. So since Mendel got 9:3:3:1 ratio, Mendel hypothesized again. And he was such good hypothesizer. So Mendel hypothesized again. And upon hypothesis, Mendel came to the conclusion that it is possible only when R and Y are segregating totally independently, and this is called independent assortment. Otherwise, what would have happened? Only these two types of gamete would have formed and these two types of gamete would not have formed, right? But since this kind of gamete could form, which was not there in any parent, it means R and Y segregation have nothing to do with each other. So in any gamete, any combination can be present, and that you can see here also, recessive R and dominant Y. And well, when you carry out self-fertilization, this big Punnett square is there. So here, dominant R, dominant Y, recessive R, recessive Y, dominant R, recessive Y, recessive R, dominant Y. And same kind of gametes here. And then you just unite these two gametes, you get this. Then this gamete, you get this. This gamete and this gamete, you get this. And this gamete and this gamete, you get this. And then what you get is 9 round and yellow, 1 wrinkled and green. These two are what? Parent type. Correct. But 3 round and green, that was non-parental type. There was no parent which has produced anything like this. Parent when it was round, it was yellow, and parent when it was wrinkled, it was also green. But now this is non-parent type. We call it recombinant type. And look here, 3 wrinkled and yellow. So in the parent, if it was wrinkled, it was green. But now you get another recombinant category. So 10 out of 16, 9 plus 1, 10 out of 16 are what? Parental type. And 6 out of 16 are what? Non-parental type, that we call recombinants. And this is possible only when all the 4 types of gametes are formed. And all the 4 types of gametes will be formed only when 2 different genes, factors, their segregation are totally independent of each other. That's what Mendel could establish. So the principle of independent assortment states that alleles at different loci, locus ka plural is loci, separate independently of one another. The principle of segregation says that 2 alleles of a locus separate when a gamete is formed. And the principle of independent assortment states that when these 2 alleles separate, their separation is independent of separation of alleles at other loci. Means RR separation has nothing to do with YY separation, Y and Y separation, or for that matter, nothing to do with T or T separation. Right? So you will get all the 4 types of gametes, and since all the 4 types of gametes are formed, so you will find this kind of ratio. So these are the 3 laws of Mendel. Dominance, first segregation, next dominance, third is independent assortment. And if you have to write the difference between segregation and independent assortment, so segregation is like each individual organism possesses 2 alleles encoding a trait. Alleles separate when gametes are formed. Alleles separate in equal proportion. And independent assortment, alleles at different loci separate independently. And later, when we study cell bio, we will be connecting it with the relevant stage of meiosis. So these are the 2 fundamental laws. Now we come to test cross. So test cross is basically a cross to determine genetic purity. Let's say that. How would you know that a tall plant that you have got is whether it is this or it is this? Correct? Because in both the cases, the result will be tall only. But this is called pure tall and this is called heterozygotic tall. All right. How to test that? Simple. You cross it with homozygous recessive plant. Homozygous recessive. So if after crossing with homozygous recessive, if it gives rise to a still tall, correct, then it is pure. And if it gives rise to 50% tall and 50% dwarf, then it meant that it is tall, but it is heterozygotic tall. Let's look at the Punnett square here. So it is heterozygotic tall, so it produced dominant and recessive gametes. And you are crossing it with what? Homozygous recessive. So look here, heterozygotic tall, but still tall. Heterozygotic tall, but still tall. But other 2 offsprings are what? Short, short. Because here also recessive allele, both the sides. Here also recessive allele, both the sides. So what do we do now? We can say that the ratio is 1 versus 1 if it was heterozygotic. Correct? So that ways you can see. And in dihybrid test cross, the same thing will be 1:1:1:1. Right. So why it will be 1:1:1:1? So simple. You can simply make a Punnett square here. So let's say that there is a 1 meter square. Okay. And take a look on the gametes. So we are looking at a trait which is heterozygotic. So let's say that it is tall, tall and round, round. Okay. So what are the gametes that you are expecting? T, R gamete, both dominant, then one dominant, one recessive, then one recessive, other dominant, both recessive. And similarly, on this side also, wait a moment, मुझे थोड़ा सा add करना पड़ेगा. So similarly, on this side also, you will be finding the same set of, no, not the same set, it is test cross. So you will be finding here the parental, here the parental genotype will be homozygous recessive. Here the parental genotype is going to be homozygous recessive. So in this one, what is happening is this. One sec. So there are only going to be two types of gametes. Actually, one type of gamete only. So the gamete is going to be with T and R. So let's say हमने दो बना दिया, but it is one and the same. Okay. Now here we are, and look at the ratio. You will get the picture. एक्चुअली वो दूसरे वाला तो अननेसेसरी ही बना दिया, लेकिन बना दिया तो चलो, वी विल सी. सो दिस इज टी टी आर आर dominant recessive for both. This is again dominant recessive, recessive recessive. This is recessive recessive, dominant recessive, and this is all recessive. And this is going to be a copy of this, so forget that. This should not have been drawn in the first place. Okay. Now let's read the phenotype. So phenotype is, this is tall and round. This is tall and wrinkled. This is dwarf and round. And this is dwarf and wrinkled. Can you see that? Same will be here also. Tall and round, tall and wrinkled, dwarf and round, dwarf and wrinkled. Can you see that? Color of the pen. अभी change करता हूँ. Wait a moment. यही एक फायदा है थोड़ा कि ये चीजें change कर सकता हूँ मैं. What color do you like? This one? Okay. So what do you see? The ratio. Tall, one sec. The ratio. Tall and round is 1. Tall and wrinkled is 1. Dwarf and round is 1. And dwarf and wrinkled is 1. So what is the ratio? Ratio is dihybrid test cross ka 1 versus 1 versus 1 versus 1, and monohybrid ka 1 versus 1. So I hope you understand dihybrid test cross also. So well, that is all about Mendelian experiment. Okay. Now there were 3 reasons for Mendel's success. Number 1, his choice of experimental material, because we have seen there are 6-7 advantages that it offers. Then the 7 characteristics that he chose, because here these 7 characteristics were very clearly, these 7 characteristics were very clearly, I would say that were very clearly producing only 2 contrasting traits, no third, and fourth. He actually avoided those traits, okay, where third, fourth trait could be seen. And then we have seen how methodical his experimental approach was. So these are the three reasons which caused success. And I have mentioned this because this is how questions have been asked. So you need to do this. Okay. Finally, applications and limitations. So in most of the cases of inheritance in sexually reproducing organisms, you will see Mendelian principles applying. But at the same time, in things like polygenic inheritance, pleiotropy is when a single gene governs multiple features. That's pleiotropy. So in pleiotropy, you will again see that Mendel's rules are not followed. If two genes are interacting, it will not be followed. Linkage and sex linkage, it will not be followed. If there is environmental and hormonal effect, it will not be followed. Okay. And then if there is codominance, incomplete dominance, it will not be followed. If there is cytoplasmic inheritance, it will not be followed. So although the list of limitation is a long list, but let me tell you very clearly that despite this so-called long list, in most of the cases, most of the traits are Mendelian traits in sexually reproducing organisms. So that's the thing. And these are the questions that you have to practice. First, design of Mendel's experiment and choice of experimental plant. Collect 15 points. Reasons for success. So everything in question number 1 plus 3 more points which I have given. So that is the second question. Then Mendel's laws, segregation with a cross, independent assortment with a cross. So about 20 points to explain plus the chart. And prepare independent assortment separately because it is an important one. It had come even the last year, 2022. So these are the questions that you should be preparing. And since we do not have much time, so itna to pakka hai ki today we are not going to do blood group genetics, but I think we still can do the next topic, that is multiple allelism, because multiple allelism is pretty much an easy topic here. So we are going to do multiple allelism, and there are just 5-6 slides so we can do. And जब हम ह्यूमन ब्लड ग्रुप्स अगले क्लास में करेंगे, दैट्स मंडे. सो जब हम ह्यूमन ब्लड ग्रुप करते हैं तो फिर वी विल अगेन बी सीइंग मल्टीपल एलिलिज्म इन एक्शन. सो मल्टीपल एलिलिज्म इज नथिंग but extension of Mendelism. So Mendel, by design, he chose only those characters which had two alleles and two contrasting traits. If there is Shraddha is asking, can you please explain dihybrid cross slide again? Shraddha, now you revise. I don't want to go in the reverse mode. You revise it and you work with the material, and then we will talk in the next class. Okay? So this is not as difficult as it appears. It's there in the NCERT also. Okay? So एक बार उस पे काम करो, and then we can. I don't mind revisiting, but not now when I have already started multiple allelism. So multiple allelism, I was telling you that what happens that Mendel deliberately chose those features. Mendel deliberately chose those features where there were two contrasting types: height, tall dwarf; color, purple white; position of the flower, axillary or the apical, so on. And as a result, there will be two alleles. But when Mendel's work was rediscovered, and when the scientists tried to repeat the same type of experiment with other organisms, it became quickly clear that in nature, there are many genes for which there are many different alleles. This phenomenon is known as multiple allelism. This phenomenon is known as multiple allelism. And why would multiple alleles arise? For this, you have to answer a simple question. Why do alleles arise in the first place? Why should a gene have alleles? And the answer is that for any species, the population is very big. And if the population is big, it is understandable that some of them will go through mutations. So, mutation in a gene may affect the functioning of a gene. And when, due to mutation, a gene's function is changed, so this changed version of the gene is an allele, of course. And if there is a big population, theoretically there can be so many alleles, isn't it? So multiple allelism or multiple allele is not as rare a phenomenon as we would like to think. There are so many organisms and there are so many cases where we see multiple allelism. In this one, we are going to primarily focus upon the human ABO blood grouping and Drosophila eye color. But let's first familiarize with this one, the basic concepts here. So mutations can modify the genetic information in many ways, correct? And therefore, for any gene, the number of alleles within the members of a population is not restricted to 2. It can be several. So when there is 3, that is more than 2, or more alleles for the same gene are present in population, it is known as what? Multiple allelism. So, but for this, at least 3 alleles have to be there. It can be more, but at least 3. So the phenomenon is known as multiple allelism. The phenomenon of multiple allelism can be studied only in a population. Why? Because a single individual will always carry at the most 2 different alleles and sometimes 2 copies of the same allele. Are we getting the picture? But at the level of a population, yes, you can basically study many alleles. You can discover many alleles. And because there are many alleles, so you can easily see many phenotypic variations. So there are many alleles, and as a result, you will see many phenotypic variations. And that is multiple allelism. First example, ABO blood types. It was discovered by Karl Landsteiner, the same Karl Landsteiner who also discovered MN blood type, RH blood type. So Karl Landsteiner, his contribution to determine the blood type is enormous. So he noticed that there are presence of different antigens on the surface of red blood cell. And a given individual can have only A antigen, that person will have A type blood. A given person can have only B antigen, that person will be having B type blood. A person can have both A and B type antigen, that person has AB type blood. And a person may have neither antigen, that person will be having O type blood. So when you study it a little closely, so what you see in this case, what you see in this case is that the alleles for this one is actually A type allele, B type allele, and O type allele. So this is the genetic basis. The gene is located on chromosome number 9 of human beings, and there are 3 alleles. IA, iso-agglutinogen, that's the name of the gene, and type A, iso-agglutinogen B, and O means 0 here. Okay. So As a result of three alleles, we see four different phenotypes. Person can be homozygous for IA and IA, and that person will have only A type antigen and the blood type will be A. A person can be IA and recessive I, still this A has expressed itself, and as a result the person has A type blood. Same for IB homozygote or IB with recessive I. In both the cases, B type antigen will be there and B type blood will be there. And here IA and IB type of genotype and both the types of antigen, and the person will have AB blood type. And a person homozygous recessive for this gene will have neither A nor B. That person will have O type phenotype. And when it comes to dominance relation, IA and IB are codominant. That will mean if both the alleles are present, both will be expressing fully and neither will be masking the effect of the other one. Therefore, they are codominant. But individually IA or IB is dominant over IO or recessive I. So this is the dominance relation. That is the first example that we have got. In your notes also, the same example is there. Second is white locus in Drosophila. So in Drosophila, which has been extensively studied by the team of Thomas Hunt Morgan in early 20th century. So in Drosophila, there are many genes which show multiple allelism. Now let's talk about white locus. So white locus is a locus that determines the eye color in Drosophila. It was discovered in 1912 by T.H. Morgan and Kelvin Bridges. They are Nobel laureate scientists. So Kelvin Bridges also established the sex determination in Drosophila, and Morgan worked extensively on linkage and linkage analysis. So white locus has got 100 different mutations and as a result 100 different alleles. And these alleles, their combination will determine the exact eye color of Drosophila. And huge variety. Out of them, some of the notable examples are these. And you don't have to, and I would actually be saying you can't and you shouldn't try to remember all of these, but maybe 5 of them you would be doing well to remember. So W allele, where the eye color is totally white, no pigment comes in ommatidia. So pure white. Then WA, that is called white apricot. So eye color is yellowish orange. White buff, so that is light buff color. Then white blood, that is yellowish ruby color. White coffee, that is deep ruby color. You pick up your favorite color. 5 of them should be mentioned. What's the mechanism? That's the last slide basically. So essentially the product of white locus is a transport protein. The product of white locus is a transport protein that will be transporting the pigment to the ommatidia. Okay. So the the fly that is white, okay, will be lacking that transport protein. As a result, there will be no pigment reaching the ommatidia and the eye will be pure white. But in all other alleles, there will be different efficiency of carrying the pigment. Not pigment will be different. Pigment is the same, but different efficiency of carrying the pigment. So while flies expressing the white mutation can synthesize eye pigment normally, they cannot transport them into the structural units of the eye, thus rendering white phenotype. And rest of them will be moving the pigment with different efficiency. As a result, the eye color will be different. Okay, so that is the multiple allelism. That is all you needed to study here.